the cell death pathway genetically in C. elegans, developmental organism. And in these studies, they defined a centrally important gene product that they termed SED3. And for a long time, they had no idea what SED3 was. It was essential for mediating cell death, and they had absolutely no idea. But one day they ran it through the databases. Remember, this is in the early 90s. And lo and behold, out came mammalian interleukin-1 converting enzyme. So then they realized that SED3 is a protease. SED3 is a protease that cleaves after an aspartic acid residue. SED3 is a death protease. And it was for this discovery that Bob Horowitz received the Nobel Prize. So this got us thinking that maybe there's a connection with our system that we are studying. Remember, we are studying death receptor-induced cell death or apoptosis. And we thought maybe death receptors engage ICE or ICE-like proteases. So in order to address that question, we collaborated with Guy Salveson, who was at Duke at that time. And he had cowpox virus encoded serpin termed CREME that was a potent inhibitor of ICE and ICE-like enzymes. So we said, whoa, this is exactly the tool that we need, because we can do a very simple experiment. We can take cells in culture, and we already know that if we treat them with TNF or anti-FAS, then these particular cells, they will die. However, much to our delight, we discovered that if these same cells were expressing CREME, this inhibitor of interleukin-1 converting enzyme and interleukin-1 converting enzyme like proteases, then behold, there was no death. And in fact, these cells grew in the presence of what was previously a lethal stimulus. So this was around the mid-90s. And what it did confirm is that downstream of the death receptor, downstream of the death domain, must be an ICE-like protease because of the CREME inhibition experiment. And by this time, there was a realization that these proteases were part of a family. They're now termed Caspases, as I'm sure you know. The C donates that these are a cysteine protease, and the aspase, this substrate specificity of cleaving after an aspartic acid residue. Now, in the subsequent next 2 years, we were able to essentially discover the signaling pathway. The first thing that we discovered was that the death domain is really a protein-protein interaction motif. And it allows for the recruitment of an adapter protein that we termed FAD, because FAD too contains a death domain. So there's a homotypic interaction between these two death domains. Surprisingly, FAD contained a second death fold that we termed the death effector domain. And even more remarkably, this second death fold, the death effector domain, was capable of recruiting a death protease, now known as Caspase 8, because the zymogen form of this death protease within its pro-domain had a death effector domain sequence embedded. So now a second set of homotypic interactions allowed for the recruitment of this death protease termed Caspase 8. And so this we dubbed the disk for death-inducing signaling complexes, and it emphasized the first lesson, which is that the death folds serve as a molecular glue. These are homotypic interaction motifs. The second lesson, which I won't get into, is that once you assemble this complex, you get activation of the zymogen through an induced proximity mechanism. And quite remarkably, this activation leads to the third lesson, that the second messenger that emanate. Once this protease is released into the cell, much like in the coagulation or complement cascades, there's an amplification reaction because the cells have other caspases that exist as inactive zymogens, such as caspase 3 and caspase 7. And this active protease is going to cleave them after an aspartic acid residue and catalytically activate them to the active protease. So now all of a sudden you have got precipitous activation of protease activity within the cell, cleavage of death substrates, and demise of the cell. All right. Now, what I want to emphasize is that there's a lot of interest in cancer immunotherapy. But the final event in cancer immunotherapy is the killing of the cancer cell by a cytotoxic T cell. And I'm going to play you a movie here where the purple cell, which contains these dots, these are actually granules, are going to enter this green cell, which is a tumor cell, and cause it to apoptose. And then I'm going to show you that this mechanism that's at the heart of cancer immunotherapy is actually a mechanism of death that is due to caspase activation. So let's see the movie first. All right. So it'll be quite quick, but I think you'll appreciate it. Bang, there it goes. So now you want to ask yourself, what happened? What did this cytotoxic T cell do to this cancer cell that just killed it? What happened? What were the molecular events? So that is shown here again in this recapitulation of this movie. And I'm now going to show you a cartoon representation of the reactions. So this is the cytotoxic T cell in purple, and this is the tumor cell in green. Now, what you're going to observe is that the cytotoxic T cell has these granules that contain two very important components. They contain perforin, that's going to form channels between the cytotoxic T cell and the tumor cell, and this protease, granzyme B. Now, remember I said granzyme B is a serine protease, but it cleaves after an aspartic acid residue. So therefore, come into motion. What you're also going to see is that the cytotoxic T cell will express the ligand for the fast death receptor, which is on the tumor cell. And this will lead to caspase 8 activation. And due to granzyme B and caspase 8 activation, you will now get two mediators, two catalytic mediators that can explosively activate downstream caspase zymogens by cleaving after aspartic acid residue. So let's just look at the movie. So there's the degranulation reaction. There's the perforin channels. Granzyme B is introduced. The ligand is expressed. You get the death-inducing signaling complex. You get both caspase 8 and the granzymes capable of activating these zymogen forms. And once you get this precipitous activation of proteases, you get rapid death of the cell. So when you talk about cancer immunotherapy, this is the reaction that we are talking about. It's this reaction, and this reaction is mediated by caspase activation. All right, so let's now introduce the inflammasome. Inflammasome has to do with the regulation of caspase 1. Now remember, caspase 1 is the original interleukin-1 converting enzyme. And this enzyme, which happens to occur in an inactive pro-IL-1 beta interferons. Now, caspase 1 is regulated by an apparatus that is termed the inflammasome. It's an upstream apparatus. Now, this apparatus is composed of a sensor, an adapter, and an effector. And the sensor and the apparatus is prominent in cells of the innate immune system. So myeloid cells, endothelial cells, keratinocytes. And it's an intracellular apparatus. So if we look at a typical sensor, that would be Nlrp3. Now, this is a member of a family of sensors termed the nodal family, and we have about a dozen or so sensors. The plant kingdom has over 200 such sensors, and they form the mainstay of the immune system in the plant kingdom. Now, these sensors have a death fold. In this case, the death fold is termed a pyrin domain, a nucleotide binding domain, and leucine-rich repeats. Okay. Now, this particular sensor, the Nlrp3 sensor, it responds to insults that damage the membrane. These could be mediators of sterile inflammation, like crystalline compounds, cholesterol crystals, amyloid beta crystals, or ATP, or it could be pore-forming toxins. So whatever institutes membrane damage is sensed by this Nlrp3. How exactly the sensing happens is not entirely clear. For our purposes, let us say it senses membrane damage, and once it senses membrane damage, it's going to couple to this adapter molecule ASC through, again, a homotypic interaction, a pyrin domain interaction. And this ASC adapter is going to serve as a bridge to Caspase-1, which is the effector, through a second set of homotypic interactions, namely card-card. So again, here the molecular glue is death fold interactions. Once you assemble this complex, this was termed by the inflammasome by the late Yerkchov. And upon assembly of this complex, you get activation of Caspase-1 to give you the active protein. And this will cleave pro-IL-1 beta and pro-IL-18. Now, I want to make a very important point that even after cleavage, IL-1 beta and IL-18 don't have a signal peptide. They are leaderless. They have no way to get out of the cell. So this is a class of very potent cytokines that are known as leaderless cytokines because they have no signal peptide. The only way they can get out of cells is for the cell to undergo a form of necrotic death that ruptures the cell membrane and allows for the leakage or the release of these mediators of IL-1 beta, important cytokines, but cytokines that lack a signal peptide. So this is really their only mode of exit into the extracellular milieu where they can engage their receptor. Okay, so now you begin to understand why the apparatus is configured in the manner it is, because once you activate Caspase-1, it isn't sufficient to proteolytically process pro-IL-1 beta and pro-IL-18 to give you IL-1 beta and IL-18, because there's no way for those cytokines to get out of the cell. You then have to allow Caspase-1 to mediate a form of necrotic death that we term pyroptosis, which will allow the release of these cytokines. Yeah. So I just contrast that if you look at death receptor signaling and inflammasome signaling, there is conservation of molecular architecture. In both instances, you have a sensor in the death receptor system, Nlrp3 sensor, it's sensing membrane damage. In both instances, you have an adapter death folds, a death domain and a death effector domain for FAD, a pyrin domain and a card domain for ASC. And then finally, you have an effector, Caspase-8 for the death receptor-induced apoptosis, and Caspase-1 for cytokine maturation and pyroptosis. So now I want to talk about a discovery we made in 2011. And for the students, I refer you to the original paper because I'm going to give a very watered down Reader's Digest version of the discovery. But it was actually quite a detective story that you will get from reading the papers. So the question we were tormented by was what is the function of all of mammalian caspases? There are a dozen or so caspases. We know many of them function in apoptosis. I've already mentioned caspase-3, I've mentioned caspase-8. And then I've also mentioned interleukin-1 converting enzyme, the granddaddy of them all. That's a caspase that's involved in inflammation because it proteolytically matures IL-1 beta and IL-18. But what struck us was that there were a couple of caspases in humans that had sequence conservation or similarity to caspase-1. These are caspase 4 and 5 in humans. And these 4 and 5 in humans is represented by a single caspase, caspase 11 in the mouse. So this was convenient because if we knocked out caspase 11 in the mouse, it would be the same as getting rid of 4 and 5 in human cells. And so we've given this sequence conservation, whether these caspases, like caspase-1, also had a role in inflammation. So in order to do that, we knocked out caspase 11 and from purified LPS into cells. We found that whereas wild-type macrophages made a lot of IL-1 beta, the cells that were on caspase 11 had a very muted response. But you could say, well, this is a very artificial experiment because you've taken one component of a gram-negative organism and you've shown gram-negative organisms have tens of thousands of components. Many of them are pro-inflammatory. How do you know it's only LPS? It could be other negative. And that's a legitimate question. But what we did was we engineered E. coli in the penultimate step in LPS synthesis. And we discovered in infections that while these E. coli that were defective in LPS synthesis could engage pathways like IL-6 generation, they could not engage the inflammation. But this is, we're around 2014 or so. And we've reached, the realization is that whether you activate the canonical pathway where the major player is caspase-1, or the non-canonical pathway where the major player is caspase 11, at the end of the day, a very important event is the rupture of the cell. This inflammatory form of cell death, inflammatory form of necrosis that we term pyroptosis, that allows for the release of these leaderless cytokines like IL-1 beta. So in order to address that question, we did a forward genetic screen. This is an ongoing screen, and it's being done in collaboration with the Australian Phenomics Facility. Mice are exposed to this mutagen ENU, which induces point mutations, and pedigrees are derived. So we've looked at over 10,000 pedigrees of mice. So these are mutagenized mice, and we are asking if the exposure to the mutagen has broken, in our case, the non-canonical pathway, the response to LPS. And we can do that by, from these 10,000 pedigrees, taking bone marrow-derived macrophages, stimulating them with LPS, and seeing if there's a defect in IL-1 beta generation or cell death. Okay, then these are recessive mutations, so it requires a fair amount of laborious intercrossing and retesting to identify the responsible mutation for the phenotype you observe. Okay, so here is an example of a screen like this. Again, IL-1 beta is on the y-axis. We are stimulating with LPS, so that's the non-canonical pathway we are engaging on the x-axis. And you'll see bone marrow-derived macrophages from the vast majority of these pedigrees. They have a robust response. They're making, thousands of picograms of IL-1 beta per mil. Now, here's a defective mouse strain, and that's the Caspase 11 mutant. So that's good. That's a positive control because that's how we define the pathway. And so we know the screen is working. But almost as muted as Caspase 11 mutation is this mutant, ENUB11. So what is ENUB11? So intercrossing and subsequent sequencing narrowed it down to this gene, GSDMD or Gesdermin D. And even though today Gesdermin D is, for those who study innate immunity, a household name, when we discovered it, there was very little known about it. Because when we went to the library, it just said it's a member of the Gesdermin family, contains an ill-characterized Gesdermin domain, no obvious signal sequence or transmembrane segment, and function unknown. And the mutation we had introduced was an isoleucine 105 asparagine. So what was Gesdermin D and what was it doing? So there are a couple of experiments. I will show you. This work has since been published, so I'll just highlight the couple of key experiments. One was simply doing a Western blot for Gesdermin D. So this is macrophages from wild-type mice. You see a Gesdermin D band. Caspase 11 null mice, you see a Gesdermin D band. And then macrophages from 2 Gesdermin D null strains, and there's no Gesdermin D band. So we are looking at the right band. We are confident about that. Now we're going to stimulate with LPS. So we take wild-type macrophages and stimulate with LPS. And look what happens. There's a diminution in the intensity of the full-length band. And there's the appearance quite prominently of a P30 fragment. But when we take Caspase 11 null macrophages and stimulate them with LPS, there is no such happening. So from this, we surmise that when cells are exposed to LPS, the non-canonical pathway, Caspase 11, is activated, and it cleaves Gesdermin D to give you this P30 fragment. And we were able to reconstitute this reaction in vitro. We could take purified Gesdermin D, incubate it with Caspase 11, and we could see the 2 prominent bands that are introduced by a single cleavage, a P30 and a P20. And when we sequenced the cleavage site, it was after an aspartic acid residue, aspartate D276. So cleavage after an aspartate acid residue is what you would expect for a caspase. But why did you need to cleave Gesdermin D? Why was this important? So turned out we first convinced ourselves that the cleavage of Gesdermin D was critically important. And in order to do that, we developed some very compelling data by generating a mouse, a knock-in mouse, in which we mutated the aspartate 276 to an alanine. So these mice would express Gesdermin D, but this mutant Gesdermin D could not be cleaved by Caspase 11. And so then we compared the response of these mice to LPS, and we measured IL-1 beta here on the y-axis. And I think, as you'll appreciate, if you look at wild-type mice, they make a bucket load of IL-1 beta. If you look at Gesdermin D knockouts consistent with our mutagenesis, the I105N, they make hardly any IL-1 beta. But here was the big surprise. This cleavage dead mutant also made hardly any IL-1 beta. So why, in responding to LPS, and by response I mean generation of IL-1 beta, do you need to cleave Gesdermin D? What is happening? So that turned out to be, the reason turned out to be that you have to cleave Gesdermin D. Because when you cleave Gesdermin D, you generate this P30 fragment that is responsible for the disruption of the cellular membrane. The evidence for that was quite simple. We overexpressed full-length Gesdermin D or the P30 or P20 fragment and simply asked whether we disrupted the membrane as evidenced by the uptake of a dye, yo-yo-1. And when you express full-length Gesdermin D, you don't see dye uptake, so you have not perturbed the membrane. Similarly with the C-terminus. But boy, look what happens when you express P30. You just shatter the cellular membranes. They become highly permeant, and now the cells stain. So this fragment that we observed that was generated by caspase 11 cleavage, this fragment is responsible for the death, and it is what we refer to laughingly as ninja. So it turns out, we've been studying cell death for a long time, that P30 is highly death-inducing. It'll kill bacteria, fungi, mammalian cells, anything in the inner galaxy, because it is so membrane disruptive. So if any organism has a membrane, which organisms do, P30 is going to disrupt those membranes. How is P30 working then? When we say disrupt those membranes, how could it be working? Well, we hypothesized that maybe what happens is that when you generate P30, it oligomerizes and forms pores in the membrane. And some evidence for this was when we took purified liposomes and incubated them with P30 and did negative stain electron microscopy, we could actually see formation of pores. And some calculations suggested that this was a 24-mer. That means 24 P30s coming together, assembling together, to give you a fairly large pore with a 15 nanometer internal diameter. So this was our work then, but the work really at atomic resolution was done by Hao Wu at Harvard and Feng Shao in Beijing. And they were working on a related member, Gesdermin A3, because Gesdermin D itself is poorly behaved for structural studies. But the conclusions are the same, and the conclusions are that in the full length molecule, P20 shields P30, almost acts like a sheath for a sword. But once you lose this P20 fragment, then the P30 unfolds to reveal these beta barrel domains that insert themselves into the membrane. And this is work from Hao Wu, that this is again Gesdermin A, this is a 27-mer pore, but you can see these beta barrel domains inserting themselves into the membrane. So I think the bottom line is that Gesdermin D kills because the P30 binds to plasma membranes, oligomerizes, and forms pores. So I think I've touched upon the inflammasome, the surprising discovery of and the fact that Gesdermin D is required for this necrotic form of cell death, and the importance of this necrotic form of cell death to allow cytokines like IL-18, IL-1 beta, IL-1 alpha to exit the cell, including other mediators of inflammation like HMGB1 and ATP. And then I'll leave you with this thought that if we could develop an inhibitor to Gesdermin D, now in one fell swoop, we would eliminate the generation of a number of pro-inflammatory elements. Okay, so with that, let me thank the people who did the work. The discovery of the non-canonical inflammasome was a collaboration with Nobuhiko Kayagaki in my department and his group, Irma Stowe, Bettina Lee, and Ofer Kornfeld, a very hardworking group. And the Forward Genetics was a collaboration with Ed Bertram and Chris Goodnow in Australia. So with that, I'll end and be happy to take questions.
Thank you, Dr. Dixit, for really taking us through the journey of apoptosis, inflammation, and decades of research. So if you can stop sharing your slides, then it will be easier. Yeah. So there are a couple of questions from the audience, then we will take from our other scientists. So one question is from Nishant. And what is its role in inflammation? Perhaps it was in the beginning of your slides, I think in the early part of your talk.
Oh, UNRX is just untreated.
Okay. Probably they were not familiar with the observation. With the, yeah. So there's one more question. What is HMGB1? And what is its role in inflammasome? And who is responsible for the activation? After that, what is the mechanism in the apoptosis?
Yeah, HMGB1 is a nuclear protein of chromatin that's important for chromatin architecture. But in dying cells, when it's released, it has a second function in that it is able to mediate